Hi, my name is Matthew Murdoff, and I'm here because of my sister Charlotte, who passed away last fall after a 3-year battle with osteosarcoma. I am pleased to introduce Dr. Troy McEachron, who will be moderating the panel Targeted Therapies. Dr. McEachron earned his doctorate in molecular and cellular pathology from the University of North Carolina at Chapel Hill in 2011. He completed postdoctoral fellowships at St. Jude's Children's Research Hospital and at the Translational Genomics Research Institute. In 2016, Dr. McEachron joined the faculty of the Keck School of Medicine at the University of Southern California as an assistant professor in the Department of Translational Genomics, primary appointment, and the Department of Pediatrics, secondary appointment. Dr. McEachron joined the Pediatric Oncology branch in 2021. He will be introducing the panel, but I wanted to mention that one of the panelists, Dr. Sweet Cordero, was my sister's oncologist, who she loved very much. And now I will turn it over to Dr. McEachron, who is joining us virtually.
Thank you very much. And first, I want to just thank everyone for attending the meeting. I wish I was there to be able to be there in person, but given some unforeseen travel issues, wasn't able to attend. But I'm just very grateful for the opportunity to at least connect remotely and still have the opportunity to moderate this fantastic session. We have a really, really great session lined up and some fantastic speakers. So this is a targeted therapies session. So we'll have 4 speakers that will have a 10-minute presentation each, and then we'll have a 20-minute question and answer session. So as an introduction, we have Dr. Amanda Marinoff. She's from UCSF. Currently, she is completing her fellowship in pediatric hematology oncology with Dr. Sweet Cordero, and she comes to UCSF previously from Boston Children's Hospital, where she worked with Katie Janeway. We also have Dr. Alejandro Sweet Cordero, who is a professor of pediatrics and distinguished professor in child health at University of California, San Francisco. He is also the director of the Molecular Oncology Initiative. Then we also have Dr. Ryan Roberts, who is both a physician and principal investigator at Nationwide Children's Hospital in Ohio, and also an assistant professor of pediatrics and member of the Translational Therapeutics Research Program at The Ohio State University James Comprehensive Cancer Center. Dr. Roberts also leads the Osteosarcoma Biology Committee of the Children's Oncology Group. And last but not least, we have Dr. Brian Van Tine, who is a professor of medicine and pediatrics in the Department of Medicine at Washington University in St. Louis, and he is the Sarcoma Program Director, Sarcoma Program Director, the co-director of the Phase 2 program, and also the co-director of the Adolescent Young Adult Program at the Alvin J. Siteman Cancer Center. Just if you can join me in welcoming everyone to the stage, we will have Dr. Marinoff speaking first, followed by Dr. Sweet Cordero, then Dr. Roberts, and then finally Dr. Van Tine.
Good morning, and what an honor it is to be here at my first Factor conference and to be able to share with you my work and vision in linking clinical and omic data as a path toward precision oncology in osteosarcoma. I don't know if my slides are they up? I don't know if there's someone who can. Well, I can talk with you. I don't know if anyone's able to help get the slides up. Here we are. To start, I do have a couple of disclosures. First, I want to make it better for children with osteosarcoma and their families. And second, I believe we can and will. Sorry, I don't think this clicker is anyone able to? Green button. Okay, I just wanted to to highlight everyone again. Okay, and we all know that we face a number of challenges in osteosarcoma, which form the catalyst for this conference today. And we're all here because we want to improve outcomes for all patients with osteosarcoma, and particularly those with advanced disease who don't respond to our standard 3-drug regimen that we've been using for the past 40 years. We have blunt, imprecise tools for risk stratification. All patients receive the same therapy up front, and we're not currently incorporating any molecular biomarkers into either our clinical trials or our clinical care. We don't have targeted therapies available as part of our standard of care, and the osteosarcoma genome is extremely complex and heterogeneous, which makes it really tough to figure out the drivers of the disease and to sufficiently power studies to really understand each individual subtype of osteosarcoma. And so we need to make order out of the chaos at both the genomic level and amid the enormous amounts of data generated clinically in the era of the electronic medical record, or the EMR. These plots here are called Circos plots, and to the left we have a classic example of an osteosarcoma genome where we have the chromosomes labeled on the outside of the circle, and each of these colored lines representing a different genomic event, most of which are comprised of somatic copy number alterations and other structural variations. And to the right is my attempt to represent the enormous complexity of the billions of data points generated clinically in our EMRs, and between clinician notes, lab tests, radiology reports, pathology reports, and then getting different EMRs to talk to one another. Well, if I'm giving anyone a headache, I apologize, and I'm working on ways to disentangle this and to really be able to leverage these data that are literally at the tips of our fingers. And so our tasks are to be able to marry the clinical and the genomic data in order to define clinically meaningful subgroups of patients that tell us how each patient will do, how their disease will behave, who will respond to standard therapy, and who needs different therapy up front that really matches their disease biology. And in the coming slides, I'm going to share some data that I think can really serve as a case study for how we can use real-world clinical and genomic data to help address these kinds of questions. These are data from a study I performed with Dr. Katie Janeway and the team at Dana-Farber, in which we looked at 92 patients with osteosarcoma whose tumors underwent clinical sequencing using a targeted DNA panel called OncoPanel. And our cohort was intentionally biased toward those with high-risk disease by virtue of about 2/3 of patients having enrolled on the GAIN study, which includes 12 institutions and selects for patients with high-risk solid tumors. And the purpose of this study was twofold. One, we wanted to define the genomic landscape using a clinical sequencing test of this high-risk cohort. And secondly, to take a look and see if we could discern any prognostic factors. And what we found here is represented in this what's called an oncoprint. And to the left, we have the most commonly altered genes in the column over on the left. And then up top, we have integrated tracks showing our patient and sample-related data. And ultimately, we saw that the most frequently altered genes here were similar to what was reported in standard risk cohorts using mostly research-grade sequencing. And so we see frequent alterations in TP53, losses in RB1, CDKN2A and 2B, and then amplifications in MYC, CCNE1, and CCND3. When we looked at our prognostic factors from a clinical standpoint, we weren't too surprised to see that the presence of metastatic disease at diagnosis was the only clinical factor associated with outcome. Histological response to chemotherapy, which is also known as percent necrosis, and often used in clinical practice as a predictor of disease, was not actually associated with outcome. And so we need to do better. And when we looked at our genomic events, we found that MYC amplification, which was present in 12% of our patients, was associated with poorer outcomes. And here are the data. In our primary cohort, up top in A and B, we see that patients with MYC amplification at a copy number threshold of greater than 7 copies, and shown by the red Kaplan-Meier curves, had a worse overall survival, had a significantly worse overall survival, which we see in A, and a trend toward worse event-free survival, compared to those without MYC amplification shown in the blue curves. And at the bottom is our next step, which was to see if this association was upheld in a lower risk cohort of patients, which was comprised of 86 patients treated at Memorial Sloan Kettering, all of whom had initially localized disease. And in this cohort, patients underwent sequencing with a similar panel called MSK Impact. And again, we see that patients with MYC amplification, shown again in red, had significantly worse overall survival and event-free survival compared to those without MYC amplification, shown in blue. So this was really the first time we were able to use a clinical sequencing test to identify a molecular genomic biomarker that can be incorporated as a risk stratifying tool in our clinical trials, and if we can validate it prospectively, into our routine clinical care. We also had the opportunity to do an exploratory analysis in which we looked at the genomic evolution across longitudinal samples in a subset of 13 patients who had multiple samples collected at different time points. And I don't have time today to go into the details of this analysis, and really the sample size is too small to say anything definitively about what amounted to a heterogeneous pattern of evolution. But the reason I included this slide is I really want to emphasize that how important it's going to be going forward to collect samples at different points in time and in space to really be able to unravel the mechanisms of metastasis and chemotherapy resistance. And when we think about developing effective targeted therapies, we really need to resolve this temporal and spatial heterogeneity, because we need to make sure that the target we're after is actually present in all of the tumors in the body, and ideally in every single tumor cell in the patient's body. So what lessons can we take away from these data, and how can we integrate it into an approach of using real-world data? Well, I think there are three main pillars, and I've here termed this the multi-ohm MYC approach, but I think it's not just limited to MYC, and I think will apply to future biomarkers that we'll uncover using integrated omic approaches. And I think these molecular features can be used as, one, biomarkers for risk stratification, two, as drivers not just of disease but of biological insights, and three, as therapeutic targets. And I'll walk through examples and some current work I'm doing in each of these realms. In terms of being able to continue to discover and clinically use biomarkers for risk stratification, we need, in this relatively rare tumor, to have institutions continue to broadly share data and to aggregate and harmonize these data with linkage of clinical and omic data. And all of this, of course, relies on ongoing banking efforts, which we'll hear about later today. I want to highlight for now on the right a few really impressive collaborative efforts that are ongoing to address these kinds of challenges, including the NIH's Childhood Cancer Data Initiative and AACR's Project Genie, and of course the Target Initiative. We also need tools such as machine learning and natural language processing that help us to really access the data that we have at our hands, and to spend less time manually digging through charts and more time figuring out how we can actually apply these data to patients. In terms of driving biological insights, the idea of bench to bedside and back again is really not just buzzwords. And as an example of that, Dr. Sweet Cordero, whom you'll hear from next, and some of my colleagues who are here today, are directing significant efforts at studying the biology of MYC. And part of this is driven by our real-world data and others that have since emerged linking MYC to patient outcome. And lastly, in terms of using clinical and omic data to identify drug targets, we are able, here's a summary figure from work done by first author Leanne Sales a few years ago, really showing how we can use sequencing efforts to define subgroups of patients with genomic alterations that are druggable, including MYC here, and to use this to drive the next generation of precision oncology trials. And so overall, I think we really can translate the challenges I initially presented into opportunities by harnessing our omic and clinical data, working toward improved outcomes, incorporation of molecular biomarkers into our risk stratification schema, biologically informed targeted therapies, and different treatments for different subsets of patients that really reflects the biological heterogeneity of osteosarcoma. And that's why I'm hopeful we will make it better. Thank you to MIB, to everyone here, to my wonderful mentors, funding sources, and the incredible patients and their families that inspire me relentlessly every single day.
Okay, well, I think we're going to move right along here so we don't run out of time. First, just want to thank Anne and all the other organizers of MIB for the opportunity to be here today, and just wanted to dedicate this short presentation to the memory of Charlotte Murdoch, who was a dear friend of many of us, and all the patients who have suffered from this disease. I want to talk a little bit today about the work that we're doing to try to develop new combination therapies for osteosarcoma, and hopefully share that and gain suggestions from the rest of you. So my lab has been very interested in the last few years in various projects related to osteosarcoma, which I just briefly mentioned here. I'm going to focus, as I said, on our efforts to drive combination therapy. Happy to talk about these other efforts if you're interested later on. So some of the key questions that have motivated the work in my lab is to try to understand how metastatic progression and therapy resistance is enabled in osteosarcoma. What are the drivers of these two phenomena, which I think are closely intertwined? And what are the specific vulnerabilities that we can derive from the genomic complexity that you heard about in the previous talk? And more recently, we've become interested in trying to understand why osteosarcoma, despite its genomic complexity, doesn't activate a strong anti-tumor immune response. And then lastly, trying to define subtypes of osteosarcoma and how we can use those to inform therapeutic approaches. So over the last few years, we've put a lot of effort into developing new models to study osteosarcoma, to really try to capture some of the heterogeneity that we all know is a sort of a critical aspect of this disease. So in order to do that, we've developed a program where we can isolate both metastasis at the time of surgery as well as primary tumors, and then do whole genome sequencing and RNA sequencing from the primary patient material, as well as graft those samples into mice and try to develop patient-derived xenograft models, which we can then use to develop cell lines to further our ability to study the heterogeneity. So we've developed a variety of models shown here. I'm not going to talk about them in detail. I'm happy to share them. We've shared these models with over 10 labs around the world already, and happy to share them so you can just reach out if you're interested. The main point I wanted to make is that one thing that's gotten us excited about these new patient-derived xenograft models is that we've made a lot of these cell lines shown on the table on the left. And what's shown on the right is called the dendrogram, which is basically transcriptional data, RNA sequencing data. And it's better seen, I think, in the next slide, where if we sort of zoom in, you can see that the trios, what we call trios, where basically we have the patient sample, the PDX, and the cell line, they tend to cluster together. And they cluster very distinctly from a lot of the kind of what we call commercially available cell lines or well-known cell lines like SJSA, E2OS, MG63.3, which kind of cluster as a separate group. And so this kind of makes us think that these patient-derived xenograft models really do perhaps reflect at least aspects of the biology of the disease that are not captured by these kind of more established lines that have been in culture sometimes for decades. So that's kind of driven a lot of the work that we're doing. This is a kind of very busy slide, but just kind of outlines some of the approaches that we're using to develop models to study metastasis. So in addition to being able to develop these PDX-derived cell lines, we can take these cell lines, graft them back into what we call an orthotopic model, where we put the tumor into the limb of a mouse. We can then amputate that limb and follow the development of metastasis either with micro-CT or with bioluminescence. And again, I'm just kind of zipping through this for lack of time. We've put a lot of effort into characterizing the metastatic potential of these cell lines. On the left here, you can see the bioluminescence signal of these cell lines implanted into mice after the mice have been had their limb amputated, and you can see that there's some cell lines make a lot of metastasis in the lung or in the liver, whereas others make very little metastasis. So trying to understand why that is, and we hope will also help us to understand the metastatic process in human patients. And on the right side, you can see also just the time to amputation, which is the solid bar, and then the time to the endpoint for the mouse, which again shows you the sort of variance in terms of the aggressiveness of these cell lines. And again, we've sent these cell lines all over the world, and happy to share them. And I think they'll hopefully be a valuable resource to the community. So in terms of combination therapy, what are we doing? So Amanda already showed this figure from a paper we published a few years ago, just kind of setting up the concept that you could potentially, and I should say we're not the only ones to think about this, but that you could subcategorize osteo and treat them based on the key driving events. And we showed in this table, also in that paper, that we were getting pretty significant responses when we targeted the agents to the drivers present in the particular sample. But of course we know that what we really need to do is drive combination therapy, but combination therapy is difficult to do for a number of reasons. So in vitro combination studies often don't reflect the in vivo response. Combination studies are difficult to do in vivo because they're expensive, and you need to think about the timing of the drug and the concentration of the drug. We also have limited knowledge about the biology of osteosarcoma, so we're sometimes stabbing in the dark in terms of really understanding what we need to target. And then there's also pressure, obviously, from patient advocates and foundations as well as physicians to move rapidly towards clinical trials. So we have to kind of move to the clinical trial perhaps before we're really ready in terms of the basic knowledge. And so we really think that one of the things we need to do is develop more fundamental approaches, including screens and animal models, but these are obviously also expensive. So the approach that we've taken to kind of start to move towards combination therapies is again using this panel of cell lines. This is a Western blot just indicating, or a series of Western blots indicating the expression level of key driver genes in this panel, or in some of these panels of cell lines, just to really just highlight again the heterogeneity of the expression of these drivers across the cell line panel, which we think, if you look at MYC, for example, or CDK4, some cell lines have really high levels, some cell lines have really low levels. And so that tells us that this panel of cell lines potentially reflects, again, the heterogeneity that we know we need to study. So we've done a large single agent screen across this panel of cell lines, and I've shown this data before, I think, to this audience. So I'm going to really focus on the following steps, which is the combination therapy. So after having done this single agent drug screen, basically here you're just looking at the responses for each individual cell line. You can see that there's a fair amount of heterogeneity. So what we're looking for is small pink dots, so that indicates a low IC50, so lower concentration of the drug kills more cells, and then the area under the curve is the size of the dot. So we're looking for drugs that have a small pink dot indicating that they're very responsive. And we see some drugs that are responsive across the panel of cell lines, and some that are very specific to some cell lines but not to others. And you're going to hear from a postdoc in the lab, Eunice Lopez Fuentes, tomorrow, who's going to done some beautiful work to try to explain some of that heterogeneity in response. But here what we've done is just basically take a backbone of chemotherapy and then take drugs from that single agent drug panel that we thought were particularly promising, combine them with chemotherapy. The chemotherapy shown on the left. The drugs that we've indicated in the combination are shown at the top. And then what we've been particularly drawn to is these DNA damage response agents like prexasertib, WEE1 inhibitors, ATR inhibitors, and PARP inhibitors, which seem to have a very strong combinatorial response across most of the cell lines that we've looked at. So here the red indicates highly synergistic, so you can kind of see that most of the cell lines showed a highly synergistic response, particularly when you combine gemcitabine with these DNA damage response inhibitors. So we've now gone on to do some validation of that initial in vitro data. This is just one example of combining an ATR inhibitor with gemcitabine in one particular cell line where the single agent response was kind of moderate, but the combination response was actually quite strong. And so we're very excited about this. We think that this potentially is a path forward to try to really define combination therapy that could be used in vivo. And now what we're doing is trying to understand what are the drivers of response. We do see responses in some cell lines, but not in others. We're trying to understand sort of what is the mechanistic basis for that. Here's another example with a different ATR inhibitor. Again, very strong response to the combination therapy. And then, of course, what we're really trying to, that data was with a sub-Q model, but what we're really trying to do is drive, responses in metastatic setting. So here is a model where we've done the spontaneous metastasis, and again we see very significant decrease in the metastatic capacity with the combination therapy, as shown on the left, and then you can see the comparison with the bioluminescence on the right, and that is reflected in the survival of these mice. So just to summarize, I think we all agree that osteosarcoma is a highly complex, heterogeneous disease with a diversity of recurrent driver alterations. We hope that these models that we've developed, that will be a resource for the community, will help us to identify better combination therapies, and through the work that we've done in the lab, we've identified some combination therapies that we're excited to drive towards clinical application. And I'd just like to end there and thank our funding sources, in particular St. Baldrick's and Battle Osteosarcoma, which have been key partners for us in much of the work that I described today, and our collaborators and the members of the lab. So thank you very much.
Thanks, Alan. The next speaker is Dr. Brian Roberts.
Thanks for having me here, and thanks for being here today. I think it's an exciting time to be a doctor and a scientist studying osteosarcoma. I think there's a lot of things happening that make me very optimistic about future progress. So in my lab, we really have hyper-focused. I've got a huge component of my lab that's just focused on what is it that mediates this process where the cells of osteosarcoma will grow in the lung, but not really in other tissues. And I think historically we've thought of this as a process where we have this big primary tumor and there's so many cells undergoing so many changes that by luck or chance or selection, we end up with this subpopulation of cells that has these incredible capabilities to survive in the bloodstream and migrate through tissues and then ultimately grow in the lung, resulting in kind of this clonal expansion of cells that we know as a metastasis. And I think the data that we and others have shown kind of brings a lot of this into question. And I think it's better that we think of these metastases really more as kind of developing organs than as growths of strange cells, because it requires the cooperation and the interaction of not just tumor cells with their surrounding tissues and environment and the ways that they manipulate both the cells native to the lung and the cells drawn into the tumor, but as I'll show you, it also requires collaboration and cooperation between tumor cells with different phenotypes. So recent technologies have let us understand a little bit better how one tumor cell is different from another. And I think previously we were really only able to look at them as a whole population, and we would see them kind of as an average behavior. And now we can kind of separate those behaviors out little by little. And this here is a UMAP plot. So in this plot, each individual dot there is an osteosarcoma cell. And within that dot are 26,000 data points of RNA expression, so gene expression. And when we go kind of try to sort those out, we use a process that puts cells that are like together and cells that are different, they spread out a little bit differently. We get a cluster plot that looks kind of like this. And as we've accumulated more and more and more and more of these tumors, we've found that there's actually a lot of patterns that are conserved across all of these different tumor types. And I'll show you in a little bit why some of this is important. But we can divide our osteosarcoma tumors into a population of cells. We call this the osteoshark plot. But over there in the head of the shark and the fin of the shark and the little plume of yellow smoke above it is a population of cells that's responsible for proliferation and growth of our osteosarcoma tumors. But over on the left are an important group of cells that are a little bit different. These cells are not highly proliferative. They're relatively quiescent, and they can be divided into a set of cells that establish and sustain the underlying matrix of the tumors, and a set of cells in the blue there that are responsible for interacting with the surrounding tissues that are inflammatory and interactive cells. And we can study these different subpopulations of cells and study how they respond to the lung environment. And we can use the changes that occur in these tumor cell subsets to kind of infer some characteristics that endow cells with metastatic capacity. And you can see there's some metabolic things that seem to affect the way that the cells can grow. So if cells are highly glycolytic, they tend to grow better within the primary tumors, whereas if they're highly inflammatory, they tend to grow better in the lungs. And as we've learned more and more, we've come to understand this, but one of the most striking things that we've found is that this heterogeneity, the differences between tumor cells within a tumor, we used to think was something that would arise within the primary tumor and would create a subpopulation that could then do, kind of like I showed you earlier, then colonize the lung, and so you would have a restriction of the phenotypes that you see within the lung. And we found that that's actually not true, and so model after model after model, if we compare the primary tumors to the lungs, you have every bit as much heterogeneity, as many different sub-tumor cell types within the lung as you do within the primary tumor, which is something that kind of argues that this these differences between one cell and another are important and may give them some kind of a selective advantage. So we've come across some of the mechanisms behind some of these differences, and this is kind of an older plot now, but one of the first papers that I published when we started studying this showed that if we blocked two cytokines, IL-6 and IL-8, so those two drugs, SC144 and DF2156A, block those two cytokines, in mice that had metastatic tumors, we could stop many of those tumors from many of those mice from developing metastatic tumors. But this was really kind of a serendipitous finding, and we didn't understand why. As we kind of started to study that further, we got some really interesting results. First of all, we found that it was actually the tumor cells that were producing the IL-6 and IL-8. We had initially thought it was actually the lung cells. And we found that it was the lung cells that caused the tumor cells to produce these cytokines. But perhaps most striking, if you look at that figure on the left, the red is a stain for cells that are producing the IL-6. And you can see that there's actually just a really small group of cells that's actually producing the cytokine. Most of the cells don't. And as you think back to the figure that I showed you earlier, these are those inflammatory cells that I told you about, whereas the other cells are doing different jobs. There's a distribution of labor here within the tumor. And we've found that these cells can be marked with another gene called P21. That seems to be kind of a module that marks these cells. And so these cells that are highly inflammatory are also not proliferating. So P21 is often a gene that's associated with cell cycle arrest. And if you look within metastatic lesions in mice, where we can kind of follow these lesions over time, you can see that these, we call them anchor cells now, are the only cells that exist when the lesions are initially established. So these cells seem to have a particular ability to kind of set up shop in the lung. But then over time, we see that they become the minority, and we didn't really understand whether that was because these cells start with that phenotype and then transition into different phenotypes, or whether this was a cooperative event. But we've started to get some data about that, and this is my favorite experiment that kind of shows how we're currently thinking about it. And what we did is we took a set of osteosarcoma cells, we divided them in half, and we labeled half of them red and half of them green. So we initially put the green cells into our mice and then waited about 14 days to kind of let them do whatever they were going to do. And then after that 14 days, we put the red cells into the mice and then waited another 14 days to let the red cells do what they're going to do. And then we harvested the mice and we looked at the lungs. And these are metastatic lesions from those mice, and what we got was a really striking result. And you would think that the green cells that were in there for twice as long would be most of the tumors, and then we would have some tiny red cells, but the opposite happened. And what we got was tiny little clusters of green cells surrounded by massive proliferations of red cells. And we've been able to show over there in the graphs on the left that these tumor cells actually interact with each other. And they interact by causing tumor cells to migrate in response to signals, and it's the same signals that were induced by that interaction with the lung. So the lung cells are causing these anchor cells to secrete factors that, among other things, ultimately recruit circulating cells that have different growth phenotypes, so that head and the fins of the shark, into a lesion that now can support their growth where it couldn't otherwise. And so perhaps one of the most chilling discoveries that we've made is that there's a lot of therapeutic implications for these models. So if we have two types of cells, one of which is the anchor cell that is establishing the lesion, and the other is the growth cell that's actually causing the growing metastasis, what happens when we give them chemo? Well, what happens is that the growth cells die, but the anchor cells remain. And you can see that here on the top. If we treat that 4-week tumor with chemotherapy, what's left at the end after chemotherapy is a little cluster of anchor cells. And so we've been working on ways to kind of target those anchor cells too, and we think that if we can kind of understand the differences in these cells that perform different functions and that have different characteristics, that they're each sensitive to different agents and different approaches, and as we learn more about these subsets, we can begin to combine therapies in ways that will ultimately stop this cycle and eradicate metastatic lesions. So I want to thank all the people who actually did this work and who provided the funding that made it possible, and I look forward to questions.
Fantastic, and this is Dr. Van Tine.
So good morning, everybody. My name is Brian Van Tine. I'm at Washington University in St. Louis, and in honor of everybody that stayed to listen for this, I'm going to drive everything to Foxo and Puma, so you get to go on your own version of a safari today. And so what we're going to talk about is tumor metabolism, and the best thing about tumor metabolism is the names of everything I get to study. You know, as we said at the last conference, you know, synovial sarcoma is all about me, making a pun. Osteosarcoma actually turns out to be all about getting to Puma. And so I usually win from a disclosure standpoint. So just in honor of breakfast and the things we had, everybody worries about sugar, but one of the things we've really discovered recently is what sugar or glucose actually does in osteosarcoma. And this is all based on a bet I had with a postdoc. And so I said, you know, we're using methotrexate. This is the serine folate cycle, and this is coming from glucose. And then this is how this story came. And I'm going to show you actually now where we are with data that's never been seen. But, you know, everybody says, well, we can just go on a low glucose diet, and therefore we could control our tumors, and this is so much more complicated because of a little organ called the liver. No matter what you eat, the tumors always see all the glucose they want. And so there's a lot of metabolic biomarkers that we're working on and that Lee Hellman's worked on, and I think clearly there's a lot of these that have some implications in the biology of osteosarcoma. And there is a lot of arginine biology, which is different in osteosarcoma than soft tissue sarcoma, and we're coming out with that soon. But really, PHGDH was not one of the better named enzymes. It's actually a key, and because there's drugs there, and we have to understand how to go after this. And so really the key dietary building blocks outside of glucose that we're going to get to and that we're building to very quickly are arginine, serine, and cysteine biology. One is redox biology, one is serine folate, which is why I use methotrexate, and the other is all involved in pathways that regulate nitric oxide and nucleotide metabolism. And so one of the things that I when we got into what is methotrexate that I had not appreciated as a clinician is methotrexate kills nothing. Methotrexate stops things from dividing, which is why then you give it chemotherapy. So you put things back into cycle correctly. But we thought, well, we can stop cell cycle. Can we do better? And so this is a way of looking at how cells grow in culture over time. It didn't matter how much methotrexate we gave, but we couldn't kill with it. And so we get into what is one-carbon metabolism. I'm going to try to keep this simple. But when glucose goes into an osteosarcoma cell, it doesn't make lactate. it actually gets diverted at pH-GDH and actually makes serine. Serine makes glycine, and glycine goes down into the folate cycle. And so you have this new pathway that I think had been underappreciated, but pH-GDH inhibitors are headed towards clinic, and this is why this became exciting. And the carbons that go into the serine folate cycle help make lipids, help make proteins, help make nucleotides, and actually lead to methylation, which is everything everybody's trying to drug. And so we did a triple-blind experiment with Alex Lazar and Billy Wang, where my graduate student did the IHC, a pathologist read it and didn't know what he was looking at and just scored it, and the bioinformatics were actually all controlled and unblinded at MD Anderson, and we looked at expression of pH-GDH in like 200 samples of osteosarcoma, and out of this came something that was useful. The overall survival and the relapse-free survival was actually worse in patients that overexpress something I can drug. If it had been the other way around, this would have been hard. But patients and most of osteosarcomas overexpress pH-GDH, and this is linked to the p53 mutations that are there. But the difference in survival was 15 years. So patients that didn't express this were naturally going to do well. Patients that do express this aren't going to do well, and it's therapeutically targetable. And so we started looking at these small molecule inhibitors, and basically, just like methotrexate, because you're going after the same biology, we could stop cells from growing. And this led, and then we looked at a second version of it. It did the same thing, stop things from growing. And so when you get into glucose being, we did a lot of tracing following this through, where does it go in a cell? And the serine biology had been underappreciated, or at least hadn't been looked at. And so then we started looking at what this did in terms of blockade. And so we could actually build up 3-PG, which is a glycolytic enzyme, showing that we were actually blocking this entrance into serine. And then if you block this, you found that serine went away and glycine levels went down, and you initiated a cell stress. But what's interesting about osteosarcoma, unlike breast cancer, where this drug's actually being developed, is that osteosarcoma, for some reason, doesn't take up extracellular serine and glycine to replace this. So you get the stress response. And so this is actually much more useful, right? So NCT503 is an NIH compound. Ray's Pharmaceuticals has a better one. And so what we do is we then started saying, well, what can we do to trick the cell actually into dying? And so then we started following, with the pathways through, and I'm trying to keep this simple because this is really complicated. And so we found that the bypass pathway was all through the mTOR complex, which is just this nutrient sensing complex, which, since we drove serine levels down, really made sense that all of a sudden mTOR was saying, well, I'm starving, fix it. And this was kind of this pro-survival pathway. So then we started looking at drugs, and so we started looking at rapamycin, everolimus, perhexaline, and then this new drug out of Advention Pharmaceuticals called ALP13MT55, which is a PI3 kinase mTOR inhibitor, hence the letters all together. And so we looked at the rapalogs and the non-rapalogs, and what was interesting is we could kill with non-rapalogs but not rapalogs. And so especially ALPI13MT55, which is being developed right now, which actually gives us an opportunity, right? So that was what came out in Cell Reports, but this is all the new data. So we actually then began trying to understand what these drugs did in the mTOR pathway. And so then I have this brilliant instructor who works for me who can CRISPR anything out. And we started looking downstream at transcription and the AMP kinase pathway and the AKT pathway. We found out that perhexaline and the AL compound did not have the same biology. And what we got into is we found that NCT503 actually altered the AMP to ATP ratio, which really triggers this enzyme called AMP kinase. And AMP kinase plus perhexaline, it would begin to trigger this apoptotic cascade, and we could kill. And then we looked with the AL compound and found we were also killing through an apoptotic mechanism. And so what is PUMA? PUMA is something you see on a safari. PUMA is also, if you can trigger it, leads to cell death. It's part of the apoptotic cascade. And so all sorts of weird things like BIM and BAX and BAC, and I don't know why they named them these things, but PUMA turned out to be the cool one. And so, of course, my lab would, of course, work on PUMA. And so what we found is we could induce PUMA with either perhexaline or AL, or the AL compound. And then we could really drive up its transcription. And this actually turned out through a bunch of CRISPR experiments to be through FOXO3. So now you've got FOXes, now you've got PUMAs. And so we have the actual transcriptional program we're using to drive PUMA. And so what we find is now we've CRISPR'd out FOXO1, we've CRISPR'd out FOXO3, we've CRISPR'd both of them out. This is all FOXO3 biology. We're following all the phosphorylations and all the signaling down. And this is a lot of things that basically said, I'm going to show you the next slide. But then we started looking at synergy. we actually have triple synergy. So if you use perhexylene with the AL compound and then a CT503, we're actually able to bring in and target this metabolically from 3 different directions. And so if we get into this in an animal model, what you see is, and this is what I like to see as a metabolic investigator, and I'm going to take a little bit more time on this slide because we're almost done. If you see, let tumors grow in a mouse, and that's seen by the blue line. That's how fast they grow out if you just do nothing. The red line, and I'm getting old and so I can barely see this, is actually when you use the PHDDH inhibitor. And basically, even though there's a statistical significance, it's meaningless, right? If I told you that your son was going to live an extra 2 days, would you care? No. But if you look at what perhexylene does, it doesn't actually do anything either. So now you have 2 drugs that when I give them to the body, the cells, the normal biology doesn't get altered. But then if you look at this bottom purple line, and it went out much farther than 30 days, we can actually metabolically target these drugs selectively by going after their metabolism. So when you go after the AMP kinase side, you see the same thing. When you go after the AKT side, you see the same thing. And so once again, you go after NCT503. When you go after the AKT inhibitor biology, there's not much there. And so what's going on right now, and this is actually real-time data, there is no C yet. This experiment's ongoing. We're actually now combining all 3 drugs in an animal model. And that's where we are right now. This is where I'm trying to translate because, I was always told that if I have something in my lab that I can't actually get into people, it's useless. So I do my best. So overall, we've managed to drive you actually from your breakfast of some donuts this morning through Foxo to Puma. I hope you enjoy the safari because I'm really trying to translate this. So thank you very much. Oh, and very quickly, I did nothing. This list of people and our funders are actually what made all of this happen. So thank you very much.
That was great. So I think right now we have a little bit of time for a panel discussion. So I'm trying to figure out how to do this remotely. So I can't quite see the audience, but are there questions in the audience?
Can you hear me? All right, thanks, Jason. Our first question from the audience.
Hi, my name is David Almert. I'm a researcher at UCLA. I just joined the osteosarcoma sort of research, and I've previously been working with prostate cancer. When I see that MYC is upregulated, when you have that in prostate cancer, it always follows AR, and so what happens with AR in the samples that you evaluated?
Sorry, can you define AR?
Androgen receptor pathway upregulation.
Oh, sorry, ER. Oh, apologies. Clearly, I've not yet looked at that biology, but I think using the patient-derived data to then drive experiments in the lab, including looking at AR receptor, I think is certainly kind of what we're after in using this kind of real-world data derived from patients.
Yeah, it's a good question. I haven't looked at it. I don't know if anybody else has.
So we have a couple more questions. I have one from mother to the panel. This first one is for Dr. Marinoff. What metrics were used to determine high-risk osteosarcoma?
Yeah, that's a really good question, and I think our definitions are fluid and heterogeneous. We sort of use it to represent patients who don't respond initially to our standard 3-drug regimen, go on to develop metastases or have initially metastatic disease. We used a proxy based on a clinical trial called GAIN, which included patients with those phenotypes.
Thank you. I have another actually 2 more questions, and then we'll go over to you. Sorry. For Dr. Sweet Cordero, ATR inhibition, how was the tumor volume calculated?
We use the kind of the standard approach to that. I don't remember the formula off the top of my head. If you send me a quick email, I can follow up with you. But it's, kind of a length times the width divided by a factor. I don't remember off the top of my head.
Thank you. And then for Dr. Roberts, how much change does a tumor make in its metamorphosis to the lungs?
So, I don't know. Now can you hear me? I can tell the there's quite a bit of changes that occur. There's changes that occur both in the relative numbers of the different subpopulations of tumors, but it's a more complicated question than that because it changes over time. And so at the time that the tumors initially disseminate, there's a dramatic difference, and the cells are completely, at that point in time, unlike anything that you find in the primary tumor. Over time, as the metastases develop, you get to have populations of cells that are a lot like some populations within the primary tumor, but there's also populations of cells that are almost completely unique to the lung metastases.
Thank you for that. And if you don't mind, panelists, when responding to questions, if you could speak closely to the microphone so everyone can hear online as well.
Hello, my name is Gina Kim from CCTDI. I have one question. I have two questions actually. So one question is from to Dr. Sweet Cordero. So when you compare the primary tumors and the PDX cells, and what is the most distinct differences in genomic profile? That is my one question. And the other question is to Dr. Ryan Roberts. So like which mechanism can be involved in survivor, cell survivor, so in quiescent cell cycle phase? That is my second question.
So if I understood the first question, it was what are the genomic differences between the primary tumor and the metastasis? Was that the question? Oh, the primary tumor and the PDX. Yeah, well, I took those slides out for lack of time, but we've done quite a bit of comparison by doing the whole genome sequencing. In general, they're very similar, both at the level of the transcription and the level of DNA. We do occasionally see some changes like additional amplifications or additional chromosomal changes, but they're very similar to each other. So, and we haven't seen any like consistent changes. I think that's what you were asking, right? Do we see anything that's always changed in the PDX? We don't see that. I think there's just, it's a little hard to know whether it's continued evolution or whether, we just selected a different part of the tumor that's what grew out. But for the most part, they are very similar to the primary tumor.
And then was your question, what is the mechanism of survival of the subsets that initially survive in the lung? So that's something that we're working on. We can define the phenotype of the cells that survive in the lung. We can define a lot of the interactions that these cells can do that others can't. And we know that if we block some of those interactions, they don't survive. So at least part of the mechanism has to be a consequence of those interactions. We're actually working on some downstream signaling that may mediate survival within those cells that actually Dr. Van Tine would be excited about. It does involve Puma. But we're really still working on exactly like at a real granular level what that mechanism is.
Thank you for that. We do have Dr. McKetron online. He'd like to interject.
Yeah, actually, I think you kind of alluded to the question that I was going to ask regarding the data from Dr. Roberts and Dr. Van Tine, kind of combining that to understand, is there a difference in metabolism between the anchor cells and these other cells that kind of make up the bulk of the tumor. And then even going one step forward, given the amount of informatic expertise that Alejandro's group has, is there any way to think about an integrated approach where you can try to, more granularly define these anchor cells from a genomic and transcriptomic approach, and then linking that to differences in metabolism between the anchor cells and the bulk tumor cells?
So I think that's a wonderful observation, and I actually would like to throw out that there's two other groups of cells there that actually are fundamentally part of tumor metabolism, which are the fibroblast stromal cells and actually the immune cells. And so this is actually a four-way competition for resources, and the osteosarcomas send out all the signals to the microenvironment that says, feed me. And they do it very effectively, and so I think it's very interesting. You have more quiescent cells with a certain metabolism that has less carbons going towards nucleotides, but then you have the more aggressive cells at the same time, and so I think you're going to have to get in with the fibroblasts in the middle and start asking these questions. What can we knock out? Which is why mainly I focus on the inside of the osteosarcoma cell at this point, because I have to start somewhere.
And I just want to say, Troy, the answer is, I think that there's a lot of biology there. I just don't know it yet. We do know and have a paper coming out soon that shows how there is definitively metabolic heterogeneity within the tumors, and within these tumor cell subsets, some subsets have at least as it appears on a transcriptional level, a completely different metabolic program than some of the other cells, but we need people like Brian that have more expertise to help us understand what that all means.
This seems like a natural segue for a fantastic collaboration, and I'm looking forward to reading the paper when it comes out.
Thank you for the question. Our next question.
Hi, I'm John Ligon from the University of Florida. This question is for Dr. Van Tine. I was really impressed by how you showed the outcomes for patients with high PHGDH expression really had significantly worse outcomes. My question is, did this track with other known negative prognostic factors, such as patients who already present with metastatic disease? And if so, I guess as a more general question, for these patients who present where the horse is kind of already out of the barn in some respects with metastatic disease being present, how does that impact how you think about incorporating targeted therapy into these advanced patients?
So that data set, to answer your first question, I am not aware of it existing. So we went through two separate data sets and validated the PHGDH overexpression was associated with poor prognosis. And this is actually the MD Anderson data set, so if anybody from there can actually comment and additional things. It's actually hard to find things within, the osteosarcoma heterogeneity that actually stand up and say hello. And that was what was interesting about PHGDH, was it's kind of a fundamental thing that, the bet that actually started was we use methotrexate, this is going to be important, so we looked, right? And it was based on what we do clinically. I think that this is, we're still at the very beginning of this, and there's a lot more to do. The second question I spaced. Can you remind me what it was? In the metastatic setting, we actually build backwards. I always think of anything you graft into a mouse as a metastatic site, because it's not a primary tumor. And so these evolutions, this is why I'm actually going after, I think we're going to have to go after tumor metabolism with at least 4 drugs to actually do it right. And so I think that that's beginning to raise its head as to, what parts we can go after safely. And so I think it's coming. And I think that continuing these conversations here, especially if anybody else wants to participate, is well warranted.
A question, John Healy. I guess to start with Dr. Robertson, before anchor cells, we've had a great interest in exosomes and preparing the local environment before the first cells. And
I
I'm
love Rosie
interested
Kaplan.
in that. And the next stage in it, are there individual cells that metastasize, or these aggregates, which then further account for some of the heterogeneity of the metastasis that you're describing? And so, will you ponder that and answer it? The other question is for the whole group, and if you are moving toward combinatorial therapies, which is desirable, how do you get, Dr. Sweet Cordero, AstraZeneca and Pfizer to work on the two drugs that you were referring to into a clinical enterprise with their own proprietary interests?
Yeah, I think that's a great question, and certainly something maybe we could get a lot of help from the advocates for. I think I'm not a clinical trial designer, but I do know that doing these trials with two drugs from different companies is very hard, which is one of the reasons why, in collaboration with Katie Janeway, we decided to go after the chemotherapy backbone plus a single agent to avoid having to deal with that issue that you just raised. And just obviously the chemo, we don't have to ask anybody for permission. So maybe that's a more viable path forward. But I think some of these combinations do require two targeted agents. And we're going to need to figure out a way to get the drug companies to work with us.
So maybe I can comment as probably one of the few adult medical oncologists as a trialist in the room. I don't find it that hard to get small companies to play together if there's compelling data and they both have money. And so I think, but there's the problem. And also, if one of them's FDA approved, it's a lot easier. Where the trials get difficult when you have two non-FDA approved drugs that haven't been put together, that aren't through phase 2, though they look like they're the right drugs, we may be 10 years ahead of where we can actually go in trial. And so, but I think it can be done. I think that there was a memorial trial that showed you can do this well, that Bob Mackey ran, where you gave gemcitabine with docetaxel. And so this is precedence in our field for good ideas and then just begging. But it can be done, and I think it can be done well.
Yeah, to your initial question about whether metastasis happens as a disseminated, so there's this discourse in the scientific community as to whether metastasis actually in reality happens as single cells that travel through the blood, or whether there's these conglomerates of cells and fibrin sheaths and things like that that elicit metastasis. And I think the right answer is probably yes and yes, that there's probably multiple paths to get to it, and what these are is these are systems that are trying to maintain a homeostasis, and the homeostasis is tumor growth. And so they're very resilient, as noted by all of our therapies, and in the terms of metastasis, there's lots of ways to get from point A to point B.
So it's an organ transplant.
So I think, in my mind, I think of circulating tumor clusters as probably accelerating that process. We have a question down here in the front.
I have a question for Dr. Sweet Cordero. With your PDX cell lines and just the PDX models in general, how similarly do they reflect kind of the patient disease course? Do you see patients that had more aggressive disease leading to more aggressive PDX models generated from their tissue?
Yeah, great question. And I think the information that we have is a little bit anecdotal. We haven't studied that sort of as extensively as we probably should. I would say that in general, well, definitely metastasis grow much better in mice than primary tumors. So the rate of take of a metastasis is probably 80%, whereas a primary tumor is probably more like 30%. And anecdotally, I would say that we have a harder time developing PDXs from patients that had disease that was localized and didn't progress. Whereas if the disease progressed, we generally have it's easier for them to take in mice. So I think there is a correlation, between aggressiveness in the patient and the ability to graft into the mice, but it's certainly not 100%. And we certainly have PDXs from patients that never progressed. So the other thing is, what we've tried to look at is whether, for example, do PDXs made from a metastasis, do they have a bigger tendency to metastasize compared to primary tumors? And that's not as clean as you might think it should be. It's a little bit variable. So we have some PDXs that came from a metastasis that don't form mets in mice at all. And we have some that came from a primary tumor that do form mets. So why that is, I think is really interesting, and maybe has something to do with some of the phenomenon that Dr. Roberts was describing. But I think it's certainly something we're interested in exploring, but I don't really have a good answer for you right now.
We have another question down here in the center.
Hi, this question is for Dr. Roberts. This is Stacy Zoller from Cleveland Clinic Children's. I'm a clinician, and so, hearing about the heterogeneity of these tumors and the different populations, and thinking about how, someday we're going to hopefully be able to translate that, as Dr. Van Tine was saying. What kind of, there is a clinical trial ongoing now with the Sunshine Consortium in rhabdomyosarcoma that Dr. Trucco is a part of, where, there is a, first hit with one regimen, chemotherapy regimen, and then a second hit with a different chemotherapy regimen. Is this something that you can see happening, in sequential, with sequentially spaced regimens, or, a different combination of 4 different treatments at once? How do you think, from what you know, that could work?
So exactly what I'm thinking, and what several of us have been thinking, and I think really at the end of the day, it's going to be our improving understanding of how this works that drives that. But we've already identified several ways that we can target, for instance, anchor cells, which is probably an overly simplistic way of thinking about this as a two cell type model, but it's a start. And we've so we've actually proposed to the Sunshine Group some ways that we might study this heterogeneity, and actually osteosarcoma really lends itself to that, because our standard of care is for metastatic disease is staged bilateral thoracotomy, right? And I think we have been slow as a field for some real reasons to kind of adopt that as a way to help us understand the disease better, but it really sets itself up for, since you're a clinician, it sets itself up for a double window trial, right? Where we can use drug A, do the first resection, drug B, do the second resection, and then be able to get those samples and understand exactly what is drug A and drug B doing to each of these individual cell types, and then use that and then go on to a clinical outcome phase where we can do that. So I'm a big proponent of that. We've been proposing that. I think it would be great to be able to kind of have a system that we use as both scientists and doctors that would benefit our patients kind of at the same time. And I think we are at a place where we can organize around some ideas like that in ways that will help our patients. I didn't get to talk much about that, but that's where I'm headed to.
Well, thank you very much. We're at time right now, but this has been a really great session. Fantastic questions. So I just want to give the presenters a virtual round of applause. I don't know if you can hear that. Thank you for the opportunity, and enjoy the rest of the meeting. Take care.
Thank you for all the great questions, and thank you to our panelists. We're going to take a break. We'll be back at 10:50 to get started.